I work in Department of Paleoceanography, Institute of Oceanology, and my main research topic is application. Like my main research interest is an application of ancient DNA and sedimentary ancient DNA studies in paleoceanographic research, in the reconstruction of past climate and past ecosystem changes. And this will be the main focus of my lecture today. So maybe let's start with the very beginning. What is DNA? DNA is a molecule that is present in every single cell of our organisms. In case of eukaryotic organisms, there are organisms that has nucleus in the cells. This DNA is present both in nuclei, but some part of it is also in mitochondria. You can see it here. In case of prokaryotic organisms, the one that don't have a nuclei in their structure, the DNA is just in the cytoplasm of the cell. And DNA just encodes every information that is needed for the formation and for living and for functioning of the organism. And also DNA is unique, it's like a fingerprint that allows for identification of an organism based on the DNA sequence. It was for many, many years, it was commonly believed that studying DNA and retrieving DNA sequences is possible only for living organisms. And it is not possible to directly study any genetic material from fossil species or from any fossilized material. This changed, and we now know that it is possible to obtain DNA from some historical archaeological fossil remains. This DNA is degraded, is fragmented, is chemically modified, what makes studying it very complicated, but still it's possible to obtain the DNA sequence. So where the DNA came from? Obviously, when I say that it's present in the cells, in the organism, it's in tissues, in soft tissues. In this regard, we can study it only from living organisms, from modern materials, but also in bones, in scales, in teeth, in every spores or resting stages of the organisms. And these are like structures that can be preserved over time, and also ancient DNA can be obtained from this. Another type of DNA, let's say, it's environmental DNA. It's genetic material that can be obtained directly from environmental sample, which is like soil, sediment, water, or ice. This DNA is quite a complex mixture of the DNA that is inside the cells of the organisms, which is so-called intracellular DNA, and also extracellular DNA. So the DNA that is outside the tissue, outside the cell, and it can be just like it can be just in the environment, or it can be binded to other particles like sediment particles, for example. So the environmental DNA is quite a complex structure. And also, in this regard, we can study modern DNA, DNA from contemporary living organisms, and as well ancient DNA, the DNA that can be preserved, especially in sediments or in ice cores or across geological timescales. So let's see how it started. The first studies that reported obtaining the DNA sequence from any ancient material was the study from 1984, when the authors obtained a DNA sequence of the quagga, which is like the ancestor of the modern zebra. That was, as far as I remember, this sequence was obtained from a piece of tissue, of skin, from specimen that was collected in one of the natural history museums. Very soon after, we got a sequence, first sequence obtained from the Egyptian mummy. And then the ancient DNA research started to develop very quickly. We had a period of great enthusiasm when there were reports of getting the DNA sequence from million-years-old materials, like the dinosaur bones, like amber, or others. This is like the most, I think, most famous failure in the, maybe not in science, but at least in ancient DNA research, when in 1994, authors of this study reported obtaining a DNA sequence from a dinosaur bone that was like 80 million years old. This was published in Science. And soon after, it appeared that it was nothing but a contamination. It was just human DNA. More or less at the same time, the Jurassic Park movie, the first part of this movie, was released. So some mean people say that the authors of the study watched too much movies, but we are not mean, so we will not speak about it more. The authors became famous, and they are still remembered. I don't know if it's the fame that they wished for, but still. But now the ancient DNA research is developing very quickly, and it's applied in many, many studies. We can study the origin of species, both modern and living. We can study the conservation, the cultural heritage, the processes of domestication, of evolution, and so on. So from many different ancient materials, we can obtain very, very variety of information about the past of species, of the ecosystems. And for example, we can study like this, maybe the most spectacular applications and the most well-known are obtaining sequences of extinct animals. It started from quagga, from the zebra ancestor, and as you may see, it develops very, very quickly. So from year to year, we have more and more sequences obtained from the remains of extinct species, including moa, for example, or woolly mammoth. This is one of the most well-known and one of the most discussed reconstruction. Now we know the whole genome of the mammoth DNA. So we have a complete DNA sequence of the extinct species. Even in 2021, I think, the colossal company announced the de-extinction project. They announced that they will resurrect the woolly mammoth. Not maybe exactly. It's they want to create like the cold-resistant elephant with like features very, very similar to the woolly mammoth. But it is like technically it is not that easy, and also ethically is very problematic because we don't know and we cannot predict how introducing to the environment the completely new species, how it can change the ecosystem. So now it's interesting. It sounds nice, but now we don't have any extinct animals coming back to us, and probably it will not happen in the nearest future. Also, another spectacular example is reconstruction of the complete genome of Neanderthals, our ancestors. Also, at more or less the same time, they were discovered and also reconstructed the DNA sequences of Denisovans. Also, another hominids living more or less at the same time when modern humans were modern humans. These sequences were obtained mainly from bones found in cave sediments. But in this lecture, because this what I said is they're most well-known, most maybe discussed, mostly presented in the media and to the broad audience applications of ancient DNA. But during this lecture, we'll focus on the specific fraction of ancient DNA, which is DNA preserved in environmental material. We'll discuss mainly sediments. So as I say, environmental DNA is a very complex mixture of DNA of any organisms that lived or present in this environment, and we can obtain this DNA directly from environmental samples. This DNA undergoes many different processes like chemical modification, microbial degradation. It can be incorporated into bacterial or viral genomes, for example. So studying this, it's quite complex, and interpreting of the data can be also challenging. The first time when the DNA was obtained directly from environmental samples was in the late 80s, when it was extracted from sediments. That was modern DNA from contemporary sediments. Later, few years later, because it was in early 2000s, there were first time when there was reported sequences of plant and animals obtained from sediments. So this, what we're talking about now, is a specific fraction of environmental DNA, which is so-called sedimentary ancient DNA, which is basically the environmental DNA preserved in the sediment across geological timescales. So when talking to geological timescales, we may ask, how ancient is ancient DNA? There were estimations that the DNA, of course, depending on the conditions, because DNA preserves better in cold conditions. So most of the records we have now are coming from cold areas. But there were estimations that it can be preserved up to 1 million years. Now we know that it can be even longer, because the oldest known up to now sedimentary ancient DNA records span about 2 million years old, or even can reach up to more or less 2.5 million years old. The first record is a record from the permafrost in Greenland, when it was possible to retrieve the traces of terrestrial vegetation and terrestrial animals. The other record that spans even more than 2 million years old is a record from sediment cores from Antarctica, where there were very well recorded sequences of diatoms. But after this period of great enthusiasm, the researcher also start to think more, maybe more careful about the methodology. The ancient DNA in general and sedimentary ancient DNA or environmental DNA studies are relatively young field of research. So we need to still work on the protocols, on the methodology, established also good laboratory practice to obtain the best record and credible records. One of the first, let's say, publications of who sum up the good practices and the kind of rules that we need to follow when working with these ancient materials was the publication Ancient DNA, Do It Right or Not at All. This is a very good advice in general, I would say, but the authors there proposed 9 criteria to authenticate the DNA, ancient DNA record. The first thing is physically isolated work area. So it means that every step of the analysis should be done in a separate space, in a separate lab. Of course, we should prepare control amplifications. So there should be negative controls at every step of the analysis, in fact, to trace the possible contaminations. It is especially needed, and we need to be especially careful when studying, for example, human genomes and human DNA, because in this case, researcher himself is the biggest and potential source of contamination. Another thing is appropriate molecular behavior, which means more or less that we should adapt our reactions, our protocols to the material we're working on. And for example, the sequences we obtained need to make phylogenetic sense. The reproducibility, this is another important point. So our results should be possible to reproduce independently in other lab using the same protocol. Another thing is cloning. This is not used anymore so much. Independent replication. So again, it's connected to reproducibility of the results. And also biochemical preservation, which means that it should be possible to somehow provide indirect evidence of DNA survival, other biochemical evidence. But this was based mainly on the studies of tissue material. Here was related mainly to studies of bones or pieces of skin and so on. For sedimentary ancient DNA, there are a lot of laboratory protocols and bioinformatic pipelines for the processing data, but they are not well established yet. There is still a lot of things to work on and a lot of things to consider, and I will tell you more about it in this part of this lecture. So here you can see the overview of the approach of working of sedimentary ancient DNA. So the first step, of course, is sediment coring and obtaining our material, the sediment core. Another step is DNA extraction. So it means isolation of the DNA from the environmental samples. So you, at the final step, you have just a pure DNA in the buffer solution. Another step is sequencing. It can be also like before the sequencing, we can also prepare, for example, polymerase chain reaction to increase the number of DNA depending on the approach we choose. Another step is bioinformatics. So the analysis of the obtained sequence data, and of course the proper interpretation. And at each step of this analysis, we can have a lot of problems. So the first step is sediment coring. There are several types of cores. There are also some possibilities and some approaches, especially some like chemical reagents that allows us to trace the contamination during the coring process. But the very important thing here is to avoid whenever it's possible, of course, it's not technically possible every time, but the perfect option is to just keep the sediment core closed and transport it to the laboratory and open it in a clean laboratory under sterile condition. And also, important step is also trying to avoid touching the lining of the core. So trying to avoid sampling any material that can have any contact with the external environment. The other step is core sampling. Here you can see a picture from our lab where we did it in sterile conditions using syringes. So the core is cut usually in two halves, and then one half is sampled for environmental DNA. The sediments obtained this way, from the sediments obtained this way, we need to isolate DNA. Here you see example of the protocol of one of the commercial kits. There are several commercially sold kits prepared for isolating DNA from sediment, and basically the extraction of DNA is just a series of washing and centrifuging with different buffers, just to remove from the sample everything that is not DNA. So finally, you can get a clean DNA extract. Of course, there is as I say, there is a broad variety of the DNA kits. You can see here, for example, some examples, some names. And the important thing is just to choose the proper one, that is most suitable for your project and for your approach. Here's the interesting study that compared the efficiency of different kits from different type of samples and the amount of DNA that can be obtained with different using different DNA isolation kits. So as you can see, the differences can be very considerable. Another step is a polymerase chain reaction, which is basically making a lot of copies of your DNA. Because one of the characteristic feature of ancient DNA material is that there's usually a small amount of this DNA in your sample, especially when when you're interested in a specific group of organisms. Because you can imagine that you have DNA obtained from your sample, and it's a mixture of a DNA of very different organisms: bacteria, viruses, fungi, algae, animals, fish in marine environment, and so on. So if you are interested in one specific group, or even one specific taxa, you can use PCR to increase the number of the DNA sequences of this targeted group. There are different types of PCRs. They can be just like, say, just PCR, simple PCR, which basically is used to increase the amount of DNA of the targeted group of organisms. There are so-called qPCR, so it's quantity PCR, which is used mainly for preparing the sample for sequencing, to quantify the samples. And also very interesting approach, digital droplet PCR, which can target even a single species and can show you the number of DNA copies. So you can also see, somehow trace the abundance of the species just based on PCR results. There are a few methodological approaches to study the community structure, the community composition. The first one is so-called metabarcoding. In metabarcoding, you just amplify equally sized fragments of the target group of organisms. So this target group of organisms can be quite broad. It can be, for example, eukaryotes or prokaryotes, or more narrow. So we have, for example, markers for foraminifera, for diatoms, radiolaria, fish, and so on. Metagenomics, in contrast to metabarcoding, is amplification and analysis of all DNA fragments. So in this regard, you can amplify and you can analyze all the DNA that you have in the sample. For target capture, you can also target specific group of organisms, but in this case, you can analyze DNA fragments independent of the size. So it's a bit different from metabarcoding, but also target specific group of organisms. Nowadays, many studies, most of the studies of sedimentary ancient DNA were based on metabarcoding. So the researchers were just targeting some group of organisms and analyze the DNA of this group of organisms in the sample. Now more and more studies is using metagenomics, so analyzing the whole community. Of course, each of the approach has its advantages and disadvantages. For example, for metabarcoding, you cannot authenticate the research because most of common methods for authentication of DNA are analysis of damage patterns or the length of the sequence. In this case, in metabarcoding, you don't see any damage because it's somehow repaired during PCR, and and the sequence are the same length. For metagenomics, for example, apart from all the advantages, you need deep sequencing to really see. So you need to obtain a lot of sequencing. That can be just costly or technically very difficult to obtain the whole view of organisms. Another step of the, let's say, laboratory part of the approach is the sequencing. Maybe I will not go into technical details too much here. The approach we use, it was called next generation sequencing. Usually in the literature now, it's called high throughput sequencing. But it refers to the same method and the same approach. And based on this sequencing, you can obtain tens of millions of sequences, so-called sequence reads, from a single library of sequences. Another point another part, like the next part, when you get your sequencing data, you need to do bioinformatic analysis. I don't want you to analyze this or to learn this. I just would like to show you, this is like the typical workflow. I want to show you that it's quite complex, quite complicated, because it contains multiple steps of analysis. First, like the multiplexing means that you need to identify from which of your samples the sequence comes from. This can be done based on the sequence tags. Like every sequence have a special label that based on which you can identify from which sample it comes from. It includes also quality filtering, removing all erroneous sequences, and of course, based on the comparison between the sequence and the reference database, you can assign it to the lowest possible taxonomic level. Of course, there are a lot of issues that can be addressed at every step of the analysis, as you on the graph, from environmental properties like the natural patchiness of organisms, for example. If you take a sample, like one relatively small sediment sample, you can see only part of the community because of the, for example, natural patchiness of the organisms in the environment. There are a lot of errors that can appear during laboratory analysis, during also analysis of the data, and in case of ancient material, we can add another part to this graph. We can call it in fossilio, for example, where we can add the damage of the natural damage of the ancient DNA, for example, or its low amount and so on, which can also create problems and difficulties in analysis of the data. So maybe to sum up this part, I hope it was not too complicated for you. This is this summary is like a take-home message for you. So this is something that I would like you to remember from this part of the lecture. So to sum up, ancient DNA, it's basically DNA that can be recovered from ancient organic materials. The sedimentary ancient DNA is a specific fraction of this DNA or specific part of this DNA, which is a genetic material that can be isolated directly from sediment sample. And this DNA is a mixture of the ancient molecule that can be within dead cells or can just be free in the environment, and also the DNA within living cells. These are both like contemporary living organisms and also resting stages like cysts, spores, and so on, which are dormant now, but can grow under preferable environmental conditions. The sedimentary ancient DNA approach can be used to detect a variety of taxa, from the simplest ones like viruses to eukaryotes, including humans and other animals. It has the potential to go even beyond the fossil records and can be preserved over very considerable periods of time, up to 1 million years. What is characteristic for this material? That it's usually highly fragmented and degraded. And this is something that we need to remember and that needs to be addressed during the analysis. And also, every step of the analysis required optimized procedures, optimized for, especially for your purposes of your research and the research goals of the project, the analysis, and also rigorous contamination control. Because in case of this approach and of analyzing DNA that is preserved directly in environmental sample, it's very difficult, let's say maybe more difficult, to trace the potential contamination than in the case of material that we can obtain from tissue. For example, if you have a bone of some organisms, like human bone, and you get a sequence of completely different organisms, you know that it's possibly most likely contamination. But if you get some erroneous sequence in environmental sample, you may not be able to trace this contamination. That's why these rigorous controls are necessary. And it is the point where I would like to stop and make a short break. Let's say 10 minutes break, if it will be okay for you. And also, if you have any questions, if you like to clarify something, feel free to ask. So we can see each other again at 10:45.
Sounds good. So any questions? All questions are welcome. A little bit quiet.
If nobody has any questions, I may have one.
Okay.
Because I was also always interested in the question with this ancient DNA from sediments. If there is some correlation of like the level of preservation of ancient DNA with the mineralogy of the sediment. So, for example, maybe when there is a lot of silica, it's better. And for example, maybe with clay minerals, I had like it may go both ways because they can absorb some molecules and DNA as well. So maybe it helps preserving, but on the other hand, they can be also like a catalyst for many reactions. So it may speed up the degradation or modification of DNA. So are there any studies like that to compare the state of the preservation of ancient DNA with different type of sediments, especially in marine environments?
This is the question I would like to know the answer for as well. There is not so many studies. I've seen only few studies that were examining the impact of the sediment, structure and sediment composition on the DNA preservation. And actually, I see some studies that clay and minerals particles that can enhance the preservation of DNA because the DNA binds to the surface of this particle, so it's like less prone to any damage. But also, I seen an article where they say that sandy sediments also enhance the preservation of DNA. But all of these were quite old studies, and recently I haven't seen any experiments or any studies investigating this topic. I think it's can be quite complicated because it requires some laboratory experiments. I think it is very hard to assess it just based on the environmental sample. So we have some, like let's say, we have some information about it, but not much.
Okay, thank you.
Okay, so sounds like another topic to be studied in future as well, if somebody is interested in.
Yes.
Any other questions? If not, we will just make this technical break. So let's meet. So you can be just connected, but please come back like 11:45. Yeah.
Yes.
Okay. Okay. Thank you.
Now, so I guess we can start another part of our lecture. In this part, I would like to focus on the application and explaining more about the possible applications of sedimentary ancient DNA approach in reconstructing past changes in climate and environment. So we'll start maybe with some definition: paleoceanography. That is studying of the Earth's oceanographic history. It's based mainly of ocean sedimentary records, and it comprises the reconstructing of past history and changes in climate, in environment, in tectonic movements, and every environmental factors. These reconstructions are based on so-called proxy. So proxy, by definition, is just a measurable descriptor for an observed variable. So using a proxy is a matter of translating everything we can find in our sedimentary records, which can be chemical components like alkanones, biomarkers like IP25 isotopes, DNA, microfossils, of course, and translating this into parameters such as temperature, salinity, productivity, sea ice coverage, and so on. The proxy can be found not only in the sediments. I usually like to compare working as paleoceanographer as a bit like crime scene investigations, because we just collect evidence, small pieces of evidence, and based on this, we reconstruct what happened in this place. And this evidence we can find in ice, in the trees, or plants, in the water, and also in the sediment. For example, the ice cores can preserve the records of in the structure, in the water, in the air bubbles. They can preserve chemical components and chemical structure of the environment. And based on this, for example, we can reconstruct the temperatures in the geological past. Here you have an example of the reconstruction. This is like very canonical for oceanography, for Greenland ice cores, and the reconstruction of the temperature during the Holocene in the last 10,000 years, based on these core records from the ice caps of Greenland. Another example of proxies found in trees or rather in shrubs. Here you have a study of Arctic shrubs. The Salix glauca, it's a willow, tiny Arctic tree from Greenland. And the authors here analyzed the growth rings of these trees from two kind of sites: unaffected site and the site which was impacted by the mass landslide that occurred in 2001 in Greenland. And they see clear injuries in the clear marks of this event in the growth rates of these trees, these shrubs actually, from the site impacted by this landslide. So it can be used as additional method for dating and for reconstructing events like that. Also, we can obtain information from water in ancient information from ancient environment, from ice in this case, but also from hydrogen isotope. It is possible the isotopic composition of the water or ice that comes from this water, based on the proportions of different hydrogen isotopes. They depend on the moisture and the process of evaporation. And based on this, it was possible to obtain a record of temperature. It is not very commonly used proxies. Actually, I've never seen much studies based on this, but it's another example. It's possible. And of course, sediments, my favorite. Sediments are extraordinary archive of past conditions, past biodiversity. We can analyze both mineral composition, chemical composition. We can analyze dry size of the sediment, and each of these parameters can provide us information about the past environments. I will not tell you more about it because you will hear more next week, and I'm pretty sure that it will be a very interesting lecture. Also, what we can find in sediments are microfossils, including, like, being the most popular ones are foraminiferans, but they can be also diatoms, dinoflagellate cysts, coccolithophores. And based on the taxonomic composition of these microfossils, their abundance, the chemical composition of their tests, we can infer information about a lot of different environmental parameters: temperature, salinity, sea ice, productivity, and so on. In case of foraminifera, I will not introduce you to the foraminifera themselves, because I believe you hear a lot about forams during your previous lectures. But only 2 groups of forams can be preserved in the fossil records, and only agglutinated and calcareous forams, so the ones that possess a test that can be preserved over time. These are the only ones that we can study morphologically. But there is a huge group, very often overlooked in the foraminifera studies, which are monothalamus forams, which have only organic lining, and they are never preserved in the fossil records. We cannot study them morphologically. But it is possible to obtain their DNA from sedimentary records. So in our first studies, like very early studies of ancient foraminiferal DNA in marine sediments, we ask a question about the match between this molecular and fossil records. Do they reflect each other? The short answer is no. For multiple reasons. There can be the question of the methodology of morphological approach. For example, small specimens or juvenile forms or propagules. They all leave DNA in the sediment, but they are very often overlooked in the morphological studies, because they are in the fraction of sediments we usually don't analyze. They are in the smallest fractions that is usually not retained. Also, one of the reasons of this lack of this match between the records can be the number of copies of DNA of several taxa. For example, some foraminifera species can have a lot of DNA, so we can obtain a lot of sequences of this species, even if when there are just a few specimens in the environment. So it can create an impression. This molecular record can create an impression that this taxa was way more abundant than it really was. But if you can see on this graph on the right, here's the comparison of foraminifera record from sediment corps from deep Atlantic. And you see this red and dark red or brownish color here. They're monothalamus foraminifera and so-called environmental plates. So these are forams that are not described morphologically. We know only their DNA sequences. And they are most likely monothalamics. So you can see the overwhelming abundance of this group in the record compared to the test stage taxa, like crotalids and textularids and so on. And this is the undeniable advantage of using sedimentary ancient DNA record in studying past biodiversity and past environmental changes, that we can detect organisms and groups that are never preserved in the fossil record and cannot be accessed any other way. In this graph, you can see the taxonomic composition of foraminifera and eukaryotic communities coming from 2 sediment cores from Svalbard. One is from the fjord, the other is from Yermak Plateau, it's an area north of the Svalbard archipelago. And in these red frames, you can see the fossilized taxa, so the ones that we can study morphologically. So as you can see, this non-fossilized fraction can make up from half or more than a half to 70% of the whole community. So studying ancient DNA can give us an extraordinary view on the past biodiversity composition, including very small non-fossilized species. So another question is, can we relate this DNA record to environmental conditions and to changes in the oceanography and environment in the area in the past? Here's the example of the study of foraminifera record, only monothalamus foraminifera. Here you can see only monothalamic taxa. From one of the Svalbard fjords that spans the last millennium, last 1,000 years. And we can see there were clear shifts in the proportion, in the percentages of the foraminifera monothalamus sequences from the medieval warm period, which was dominated by the Toxisarcom and Clay P sequences, to the onset of the Little Ice Age, dominated by the Batesiphon sequences. This is this guy. And also Hippocrepinella hirudinea, to the transition from the Little Ice Age to the modern warming, dominated by this sequences, Velaria pellucidus, and the modern warming, which is dominated by environmental clades and clay degrade. So you can see that there were shifts in the community that were most likely driven by the environmental changes during the last millennium. Also, we can go deeper in this record. Here you can see the example of the record of allogromids. This is one of the groups of Monothalamus foraminifera, like the one of the example is here. These are these tiny guys. This is the cord from Storfjorden spanning the last 3,000 years. And we see clear shifts between the clades of allogromids that correlated with the periods of the more intensive inflows of Atlantic water. There are very short-term intensification of Atlantic water inflow, which created shifts in the water masses in the area, and of course, shifts in environmental conditions. And it was reflected in the composition of the allogromid community. We can go even a bit deeper, from group level to the clade level and to the variability inside the taxa, inside the species. Here you see an example of the study, another study from Svalbard. There is a record of planktonic foraminifera Neogloboquadrina pachyderma, very commonly used in paleoceanographic studies as an indicator of water masses, also very often used for dating of sediments, establishing the chronology of sediments. And we find that in this record, there were several genetic variants, so-called amplicon sequence variants, here abbreviated to ASV. Here you see the structure, the secondary structure of the DNA of this taxa. And these ASVs will differ between each other by substitutions of these nucleotides. Like here in colors, you can see that the bases of DNA in some cases were different. But in one case, for example, in ASV, that was this one, this ASV 18, there was quite a huge change. It's made some small for you, but in DNA, it's quite a big change where the double C, two bases were replaced by a single A. And the occurrence of this genetic variant coincided with the end of the last glacial maximum. So this is the time when there was very serious reorganization of oceanographic conditions in the area. That was the time when Svalbard-Barents ice sheet break up, when the Fram Strait opened again. So it allows the water exchange between the Greenland Sea and the center of the Arctic Ocean. And that probably, most likely, caused the shift in the population, genetic structure of the population of Neogloboquadrina pachyderma. And this is the kind of information that we cannot obtain using morphology, because all these morphological species were basically more or less the same. Also, not only foraminifera were used as a proxy. Here you have an example of the study based on dinoflagellate cysts. And here the authors used the digital droplet PCR approach, which can show you not only the presence of the species, but also the amount of their DNA. I mentioned this approach in the first part of the lecture. So here you can see the results of this approach. And you see that there was increase in the abundance of sea ice associated dinoflagellate Polarella glacialis in the period of seasonal sea ice cover, which is favorable for growth and development of this taxa. Also, there were some authors tried to correlate diatom data with environmental conditions and development, Holocene development of environment. But most of the study were based just like a simple comparison of proxies. So we analyzed like well-known, well-described proxies, well-established, and compare them to genetic records and just see if there is a match. If there is a match, we win. That's great. If there is not, we try to explain it. But to develop a proxy, we need quite certain information about the correlations of the presence of the occurrence of the taxa and specific environmental conditions. So then we can use it as an indicator. So here you have some examples of when we test the correlations between the proxies and between the specific environmental conditions, like the sea ice, sea surface temperatures, ice rafted debris, which can be indicator of ice rafting, so the glacial activity also. And the red color show a strong positive correlation, blue color shows strong negative correlations. So we can see that we have both groups, here we have taxonomic groups, or genetic variants, because here are amplicon sequence variants that can be correlated, in some cases very strongly, with very specific environmental conditions. Of course, this is something that needs to be tested. This is our ongoing study from Svalbard. But this is like the next and very necessary step in developing and in applying sedimentary ancient DNA as a proxy, as a regular proxy. Of course, on this way is a lot of challenges and biases. They can appear from ecosystem dynamics. They can appear from the method of DNA isolation, of amplification of the methodology during the sequencing and bioinformatics. Here I will just show you a few that are, for me, now most challenging. The first one is the using of the method, metabarcoding versus metagenomics. So the approach that is targeting specific taxa, and when we amplify, so like we increase the amount of DNA of our targeted taxa, and metagenomics, in which we amplify like the whole community. And here it's the example of the study. The shotgun sequencing is metagenomics, amplicon sequencing is metabarcoding, is another name. But you see that it targets the same group, diatoms, but the taxonomic composition was pretty different. And also there were only 10 taxa that were shared between these both approaches. So we need to carefully choose the approach for the purposes of the study. Another problem is the diversity decrement through time. It can be degradation, because it's also a very characteristic feature of ancient DNA that the older sequences, like the older the DNA, the more degraded it is. So it's it's more complicated to obtain the sequences, and it's more likely that the sequences will be like not of good quality, and only the part of diversity will be presented in the record. But also it can be effect of our primary length. So the length of our targeted fragment, because if the DNA is short, so it's obviously easy to amplify and to obtain a short fragment. But if we choose a longer DNA fragment, if we would like to amplify this longer fragment, it will be more likely that we have way less sequences in the oldest parts of our records. And for example, here you see the record of foraminifera and eukaryotic community based on two different markers, and you see that this is a mess. This is a similarity analysis, NMDS plot, and this is like just a mixture of sample. The record seems very random, and it's probably the effect of could be the primary length, could be the fragment, but it creates the appearance of a high number of so-called singletons. So these are taxa or amplicon sequence variant that appeared in very high numbers. We have tens of thousands of sequence reads, but only in one sample. And then in another sample, we have tens of thousands of reads, but of another taxa. So this creates a very random community, and it creates record that is very hard to interpret, because we cannot be sure, and probably it's not a natural variability in diversity. They're not natural changes, but rather technical biases. Another question, very important, is how well this marine diversity is recorded in marine sediments. So it is especially in regard to planktonic taxa. So because DNA of these taxa, there are groups, there are organisms that live in the water column, they develop there, and to be recorded in the fossil records, they need to sink. Sometimes it's thousands of meters. So in a marine environment, so this DNA can undergo a lot of processes on its way to the bottom. And we tested this. We analyzed DNA from water column, from surface water, 100 meters, near bottom water, and in the sediment of the same organisms. And what we noticed that some groups like diatoms, which are quite big, they have heavy tests, they are very, very abundant in the environment, they are well represented. So we see a lot of them in the water column, but also a lot of them in the sediment. But for example, for small groups, like Picozoa here, they are very tiny, they have single-celled organisms, they have organic lining, and they are very abundant in the water column, but they nearly disappear in the sediment. So we see that this taxonomic composition is a bias, is slightly shifted towards the some taxa, and this is something we need to keep in mind when interpreting our data and interpreting records. This is also the case for planktonic forams. You see here these red colors. This is the abundance. So planktonic taxa like Globigerina uvula was very abundant in the surface water at 100 meters water deep, but nearly absent in the near bottom water and in the sediment. And also we see that this is the taxonomic composition of forams from water samples from different area. So we also can see that it not only differs from the surface to the bottom, but also it differs geographically. Different colors here mark different locations, and the composition was pretty different. So this is also another point that we just need to consider when analyzing and interpreting records. Another point we need to keep in mind when obtaining our sedimentary DNA data and trying to interpret this as a proxy is the assignment. Because as I mentioned before, the identification of sequence, so the assignment, is based on a comparison of our DNA sequence with the reference database. Seems very easy, but it's not that easy, because the first problem is that the many references databases are just incomplete, especially in case of small taxa, like the single cell organisms that were not broadly studied until now. Now there are many efforts, many projects and programs collecting samples from the world oceans and sequencing them. So these reference databases are still updated, they are still growing, but there is still a lot of gaps. But also there are sometimes problems with identification based on the tool, the bioinformatic tool we use for this. Here you see the example of the study when the authors use these are names of the tools to identify the sequences. And you see that the number of identified sequences was very different depending on the tool, and it did not exceed 50% in any case. So it's not that much. And here, oops, I'm sorry. And here you see the effects of using, because in this study, authors design like so-called signature. They search for genetic signature of each forum groups. And based on these signatures, they can increase the number of assigned sequences very considerably. In inner cycle, you have the results of this research assignment when around 50% of sequences remain unassigned in this gray. Here in this outer cycle, in purple color, you see the sequences assigned by signature. So you see that this proportion of unassigned sequences decreased considerably, and we can obtain way better and way more credible record that is way easier to interpret when we know what we have in our sample. So when it comes to developing the genetic proxies of past environmental or climatic conditions, this is one of my favorite graphs. We came from the extra optimism and this joy of having DNA in a sedimentary record and this huge potential of it, then to the pessimism when we face our problem, and now we can say go to realism phase when we are aware of the limitations of the method, and we are more aware of the problems that need to be addressed, and we do address them. I would say in the case of DNA, we quite quickly come to this realism phase because there is the number of studies addressing these issues and many other issues, and exploring the potential of the DNA record is increasing very, very quickly. So now there's a huge effort in the scientific community to develop, to validate the protocols, to develop new protocols, to obtain as good records as possible. So what I would like you to remember from this part is that that sedimentary DNA, this is I think the biggest advantage of this approach and of this method, that it provides the access to the hidden diversity that cannot be studied in any other way, that are never preserved in the fossil record, but can be ecologically meaningful. It can provide us information about the past climates, past environments, past biodiversity, and the populations. Now the paleogenetic approach is more like complementary for other proxies and other paleoinvertebrate records. So it can be used to validate, to refine, to provide maybe more detailed insight into the past changes, or support other proxies. Let's say this development in using the ctDNA as an independent proxy, it's still in progress. We're still developing it. So now it's rather a complementary source of information. And before it will happen, there are a few issues that have to be addressed. The first is authentication of ctDNA records. So we need to be sure that this is really ancient DNA. This is really represents ancient biodiversity. The optimization of our protocols, of our data handling, the reference databases, and the completeness of the databases, or the establishing the correlations between the environmental parameters or other proxies and our records, and studying the taphonomy of DNA. So the processes that DNA undergoes in the water column and in the sediment that can affect the composition of the structure of communities and the composition structure of our records. And this is the point I would like to stop again. I have one short part of my lecture, so maybe we can have like 5 minutes break. Is it okay for you? Just to take a breath. If you have any questions here, again, feel free to ask. I have one more part, very short, about the molecular evolution of foraminifera. So we can see each other in 5 minutes, or if you have any questions, it's time for you.
So,
No questions.
Questions are welcome.
It usually, when there's a question, it usually means that everybody understands everything or nobody understands nothing. There are two options.
Right, as usual. Right. Yeah, if we don't see, maybe we will have a bit more questions afterwards.
Mm-hmm.
So one option is to meet in 5 minutes, or if you like to, like continue, there is another option. So it's just up to you or up to everybody. It's just
so
what you prefer.
Let's say who needs a break, because the last part is pretty short, so it won't take much time.
Yeah, we don't see anybody. So Joanna, if you can continue, then
Yes.
fine. If you need a few minutes break, then we make a break. It's just up to you.
No, okay. So I can continue to the last part of this lecture.
Great.
Yes, maybe I will start this part. Before we come to phylogeny, I will go to a bit more examples of the study DNA applications, and again start with some research challenges, key research challenges that we are facing now. The first is something I mentioned before. That the limitations of interpreting the signals, so the distinguishing between ancient and modern DNA, because I said sedimentary DNA is a mixture of both, and also distinguishing between that of alive organisms and interpreting them in context of ecological changes. Another is the understanding of past evolutionary processes that lead to creating the contemporary composition of microorganisms. And difficulty to reconstruct interactions and the networks in the environment, in aquatic environments, for example, between bacteria and eukaryotes and host-parasite interactions. Because one of the features of the DNA records is that, for example, that we have a lot of parasites. I see this in our marine sedimentary records, that, for example, we have a lot of dinoflagellates. This is one group Syndinyales that are parasites. So we have a lot of parasitic taxa, and we don't have established yet the host-parasite interactions. So we cannot really interpret this record, but they can be meaningful because they are sometimes very, very abundant. They can make up like 20%, 10%, or even more part of our records. So the first thing, this will be very brief. There are few protocols and few possibilities of distinguishing between between the dead and living assemblages in our sedimentary samples. They include both laboratory procedures during laboratory analysis and also bioinformatic strategies and bioinformatic protocols that can be applied after the sequencing to our sequencing data. Here's the one example of the study when the authors prepared independent extraction of DNA that is extracellular, so the DNA that is in the environment, and the intracellular DNA, that the DNA that is also in environmental sample, but it's within the living cells, it's inside the organisms, and you can see that the abundance of DNA of this extracellular eDNA and iDNA, intracellular DNA, is pretty different, and also the taxonomic composition was pretty different, so we can nicely distinguish these two communities in the same environmental sample. Another point on the chart is evolution of genomes. So the analysis of the genomes and reconstruction of genomes, also based on environmental samples, allows us to reconstruct the occurrence, the timings of evolutionary events that lead to the present organism, present biodiversity, and understanding the genomic changes that are leading to adaptations, to also to creating the contemporary environments. But there's also a problem, an issue, that this damage and fragmentation, which is quite characteristic and typical for this sedimentary ancient DNA, can be very problematic when it comes to assembly of genomes. And there is like the reconstruction of genomes of organisms based on sedimentary records is in its infancy, but there was one study when authors succeed to reconstruct the chloroplast genome and the full genome of algae based on the sedimentary DNA records from Scandinavia. And another point is evolution of past ecosystems. So there is a question mainly of the approach again. Whether sequence the whole community, which can we obtain a full range of organisms, but usually in this approach, most of this DNA that we obtain originates from bacteria or archaea. So if you are interested in bacteria, archaea, it's a perfect approach for you. But if you are interested in eukaryotes or even specific group of eukaryotes, like animals or terrestrial plants or algae or any other group, they make up only a very, very tiny fraction of the DNA record. So it will be very complicated to obtain a good record of this specific group. So in this case, it's better to use the targeted molecular approaches or search for a combination of the sequencing of the whole community and targeting the specific group. And that's the example of this kind of study when authors develop the kind of probes targeting a specific group of organisms. Here there was phytoplankton and specifically harmful microalgae from Tasmania. And if you see this, these are plankton baits, refers to this planktonic probes. Here are these harmful bacteria probes. And you see that it increased the amount of DNA in using these probes, increased the amount of DNA and proportion of DNA in the samples. So it was really efficient in detecting the group of interest. Here I'll show you this few examples of the study using this sedimentary ancient DNA approach. This is one of the earliest studies, one of the first studies from 2009, reconstructing the record of dinoflagellates, especially Polarella glacialis, sea ice-associated dinoflagellates from Antarctica. And the authors noticed, based on the DNA, the shift in the community towards colder climates and more extensive ice cover. Here's another example of the application of sedimentary ancient DNA record. Here's from the lake course from Tibetan Plateau, which is a reservoir for terrestrial vegetation and land development. And the authors reconstruct the terrestrial plants community in the last 17,000 years. And noticed noticed clear changes in the community from ice-free area dominated by alpine meadows to the time when the forest invasion starts. Another example of terrestrial ecosystem development is from Iceland, another based on the lake core and sequencing on the lake core. And the authors noted a clear change from the massive migration of the new species after the glaciation, and also another huge modification was at the time of the human settlement and the area when the human activities start to change and modify the environment and the flora and fauna in surrounding areas. Here's the example of the very nice, very interesting reconstruction of here's the combination of the analysis of sedimentary ancient DNA record and applying this record in creating biodiversity, in creating indices of the ecosystem state. Because most of these indices nowadays are based on the morphological studies, and the analysis of the morphological analysis of the species composition in the area. And based on this, there are indexes created. Here, the authors applied genetic data in the Gulf near Naples in Italy, in the very industrial area. They reach with the records till the mid-19th century and show the clear development from unpolluted environment to the increase on the level of pollution in the environment associated with the development of industry in the area, to extremely polluted environment with severe reorganization of the whole communities, of the whole ecosystem, to the modern reduction of contamination and restoration of the ecosystem after the actions protecting environments were undertaken in the area. Here's the example of the reconstruction of permafrost community from the permafrost sediments. Here, the microbial communities. Also, it was possible to assemble genomes and also to detect the genes, specific genes that were responsible for specific traits and specific life functions of the microbial communities. Also, it was possible to reconstruct the marine ecosystem shifts. Here, I don't want you to interpret this graph. I just want you to show the diversity and how many different groups we can record in sedimentary record. This is the record from the Arctic, from sediment core. Here are presented only planktonic organisms. In this article was another graph, the same kind from benthic organisms, and the authors could reconstruct the transition from like sea ice dominated late glacial ecosystem to more mild, more moderate ecosystem characterized by the presence of different organisms, in this case cyanobacteria, salmon, and herring. And also they will try to make some conclusions for the future based on these records and on this data, how the food supply, how the food webs changes, can modify the environment and the ranges of the species that were present in the area. As you may see, that sedimentary DNA is an emerging tool. As I mentioned, is the number of studies, the number of applications, the number of both technical and ecological studies based on sedimentary DNA is increasing. Here you see graphs showing the number of articles published in the recent years. Here is the graph regarding type of sediment. It's limnic marine or both limnic marine. Here you see the type of paper and type of groups. So you see that it's gradually increasing, and it will be increasing more. So what I want you to remember is that the ctDNA can allow for a lot of different applications. We can study different levels of communities, like from genes to genomes to populations to whole ecosystem in the extended time scales, and reconstruct a lot of past evolutionary events. That analysis of this DNA from marine lacustrine and also terrestrial environments, that are marine permafrost, can document the ecosystem of evolution of ecosystem, post-glacial changes, extinction events or invasion of new species, and also the impact of anthropogenic impact of ecosystems. And the last part, I would like to tell you a bit more about a bit different aspect of analyzing DNA. I would like to tell you how looking into modern living organisms, into single cells, how can we look back in the past in the evolution? I will focus on foraminifera, because I believe for most of us is the main group of interest. So analyzing the sequences of modern foraminifera, we can study the evolutionary relationship between the organisms, between the foraminiferal taxa. Also that can be determined by these relationships can be determined by phylogenetic inference. So it's mainly the statistics, the tools, computational tools applied to create this kind of phylogenetic tree showing the relations between the organisms. Also some heritable traits, many DNA sequences, and also morphology. So combining it together, we can reconstruct the evolution of the species and of the taxa. Here you see one of the earliest, a pretty early phylogenetic tree of foraminifera, placing it in the between, very close to some amoeba or amoeba look-like organisms. But the very most typical and the earliest this morphological based view on the evolution of foraminifera is that the foraminifera evolved from this simple monothalamus forms to agglutinated and then to calcareous tests, the most developed ones. But this was based mainly on, obviously, only on morphology. So we can observe in this case test evolution only. When we include molecular tools, apart from the test architecture, we can trace the evolution of the soft parts, like for example, reticulopodia, and genes coding the proteins and that can lead to creating reticulopodia or any physiological traits of foraminifera. So a few years later, it was published a phylogenetic tree of forams themselves. And from this, we can make a few conclusions, a few observations that, for example, Atalamida are naked foraminifera. This is, for example, the taxa reticulomyxaphilosa, shown here, that was for quite a long time considered amoeba or some amoeba-like organisms. Now we know that it's just a naked foram. Another finding was that Alagromida and Astorrhizids are from common assemblage. And also and the third point was that milioliths most likely evolved independently from other both agglutinated and calcareous clades, which was not very, very sure. Here is a few years later, that was another refined phylogenetic tree. Of course, I don't expect you to analyze it in detail, but what we can get from this, what information can we achieve? So the first thing is that multiple origins of agglutination is foraminifera. So it means that agglutinated forams evolve independently several times during the evolution of this group. The same applies to multi-chamber foraminifera. They originated at least twice during the foram evolution. Also, textularids form a series of independent lineages. They most likely, this group and the division of this group is based should be not based only on the text morphology and the structure. Another thing is the common origin of rotalids, ulimids, and globigerinids. So probably there's no evidence to separate these groups. They probably have a common origin. And also another thing is what was observed also in areas in the previous studies, that milioliths and spirulinids evolved independently. They very often also branch together in the phylogenetic trees. So you may see that we have very detailed insight based on the genetics, on the evolution of the taxa. And we need to wait a couple of years, but based on this data, there was proposed a new classification of foraminifera. Here in this table, there are classical classifications based on morphology only, but this new classification, apart from morphology and morphological trends like chamber shapes or the growth or the shape of aperture of the foraminifera, includes this phylogenetic tree and this DNA records I presented you before, and this resulted in the proposing of dividing foraminifera into three huge groups. First is the class Globothalamia, which refers to multi-chamber species with globular chambers. Tubothalamia, which is also multi-chambers, but have tubular chambers. And also the assemblage of monothalamids, all these single-chamber taxa with both agglutinated and organic walls. And what is important and interesting is this new classification that is not anymore based simply on the test structure. So it's not only calcareous versus agglutinated forams, because these groups, all of these groups, contains both. So what is important here? that, as I say, molecular studies challenge the traditional views of evolution as systematics. And it was in the contrast of what was believed before and what was usually observed based only on morphology, that not the transformation from organic to agglutinated and then to calcareous was the most important event, because this occurred independently several times during the foraminiferal evolution. But from the evolutionary point of view, probably the most important step was the transition from a single-chambered to a multiple-chamber test. So that's all for me from today. Thank you for your attention. And again, if you have any questions, feel free to ask.
Yeah, now this is time to have lots of questions, I hope. Well, Stefano is not connected. He couldn't join us, so this is that Italian student. He was always asking questions, so I hope a few of us can kind of help and ask. I guess with young samples, like we have some PhD students which are actually associated with the Institute of Geography in general. So you have pretty young subfossil record of several things. I think that could be a very useful method. So you can now apply it, or you can collaborate with Professor Pawłowska or with others, but you know where to go.
Yes, absolutely. Feel free to contact me. We have a laboratory adapted to studying ancient DNA, sedimentary ancient DNA records, and we are very open for any collaborations, any project. So of course, feel welcomed.
If you still think about questions, I just maybe mention that this drawing is actually made by Ivan Volsky, so by our Russian friend who had to leave Russia. I think he's in Georgia now because he was very much against the Putin system. So well, thank you, Joanna, for this picture. But yeah, that is the history going on around as well. Yeah, but questions are welcome. Well. If not, I just have at least one question that if you observe what is going on in paleogenomics, do you see similar trends in proteomics, in paleoproteomics? So I don't know how you call it, this field, because you probably could somehow combine these methods in the fossil record as well.
Yes, of course. But when I was doing research to this lecture, I see also some studies, very few studies, that the authors were try to reconstruct genomes, or looking for some protein coding genes, for example, or looking for some proteins. But there is not that much studies. I see that nowadays, like, the most trends are towards looking for the communities. These are the kinds of studies I showed. The researchers looking for the community changes, for the ecosystem changes. There is more and more studies looking also for the environmental impact assessments. So all this biodiversity, not biodiversity, but these indices of the state of the environment and the humans and human impacts of the environment. So also this many of the technical efforts and this study refining methodologies also are developing this direction.
Okay. Yeah, you at the end presented these phylogenetic options, and that is for me that was a big discovery. That was when we started to work on it with Jan Pawłowski and Maria Holzman, both that time from Genève. Now Jan is with us in Sopot, but there is for people working with the fossil material, this is really an amazing method, because if we still have these creatures alive, then we can really go very deep and study and test our hypothesis based on the fossils or morphology or texture or whatever. And quite often we see that we were wrong before, and we see completely new trends. So it can be applied for any kind of organism. So it's an important message for all of you, so that if still something is alive and you want to study something in the fossil, you have a molecular method now, and it is well established. So for ostracods, for radiolaria, for coccolithophores, all these things can be studied, and I'm sure they are studied now. So be open and don't trust just fossils or something what is well preserved. It could be even molecules. If they are preserved, they give new information.
And also it applies not only to organisms that are alive, because I showed that at the very beginning of this talk, that, for example, for human hominids or for some extinct species for which we have the whole genomes, we can make a phylogenetic tree as well, and we can have insight into the relationship between the modern living taxa and this extinct species, and see where there was origin of this species and how it relates to contemporary organisms.
Right, sure. Is there any chance to somehow, well, that might be a difficult thing. So if we have in Paleozoic something which is now gone, like conodonts, yeah, so little phosphatic teeth, and people working on extinct organisms from the deep time. Is there any chance, based on, like parts of the sequence, to reconstruct some parts of phylogenetics, or that is too far? I mean, like millions of years. Really,
well, there
really
were people who tried to study dinosaurs.
Okay.
And how it ended up.
So step by step, maybe we can.
I probably, I guess not. I doubt, really doubt that it would be possible, because probably it's just too old. Like the oldest known records do not expand like 1-2 million years old, and I think we are quite close to the limit. We are, of course, extending this limit. It goes beyond this limit for all the time, because the first records when reached like the Pleistocene, there were for like this hard geologist, it's pretty young sediments. So now we're going back in time, but for something that is like really fossilized from some remains that you come from the rock, it undergoes the genetic processes. It's probably not possible to obtain a credible DNA sequence. But maybe in the future we'll see what.
Yeah, I think that DNA may be impossible, but I think in conodonts sometimes some proteins, especially collagen, can be somehow preserved. So maybe it would be possible to sequence collagen sequences from it and compare it with other animals.
Mhm.
And then
They should give some information. Yeah, absolutely.
Maybe.
Absolutely. Yeah, people have record from the really old stuff. Conodonts, yes, but definitely they have good records from the Triassic. I can't remember what was that, but it was definitely the Triassic collagen, pretty well preserved. So there is a big chance to get something much older.
Yeah,
Sounds
and
good.
molecular phylogeny doesn't have to be based on DNA. It can be based on proteins as well. So that would be also molecular phylogeny.
Yes,
Right. So that would be that paleoproteomics. Yeah. So that's something which is studied. What what you, Joanna, already, yeah, that was my question as well. But that is still people are that is maybe not so well developed like studies based on DNA, but yeah, people are working on this. Yeah, so the last chance to ask 1, 2, or 3 questions.
I may have one question. Well, I could
Okay.
have more, but I will
Right.
ask maybe only one. And
Okay.
it will be similar to my previous question, a little bit, because when we have this ancient DNA in sediment, well, how can we be sure that well, it doesn't move in the sediment so like vertically, like by diffusion or something? So is it staying in the same layer or not? So yeah.
Cannot be sure. Basically, because of course there is a DNA leaching process, so it can migrate with pore waters up and down, and also considering the fact that this DNA we find in sediments, especially in the upper part of sediments, like upper 10, 15, 20 centimeters, we have also living organisms there. So there is it is a mix of this ancient and like dead fraction and the mix of this living fraction, and these organisms also migrate up and down. So they can also transport DNA, not DNA that they contain, their DNA, but also other DNA. By accident. So of course there is some degree of mixing, but of course we support our records with other proxies. That's the first thing. In paleoceanography, you can never rely on one proxy only. It's always a multi-proxy approach when you analyze different fossils, different chemical compounds, and different properties of the sediment core, and then put it back. When put it all together, then we can make a conclusion. And also in the DNA records, we usually study the most abundant groups, the groups that make like the considerable part of the DNA records, because we can assume that these were the groups that were important for the ecosystem, and they were an important part of this biodiversity. So even if the part of this DNA was transported, it is not very likely that like most of this DNA will be transported as well, disturbing our record in a very considerable way.
Yeah, and when you started talking, I thought about one more thing, and maybe it would be possible somehow to do in some experiments because if some of the DNA is absorbed on some minerals like clay minerals or something, and some is just in the solution in porous water, maybe it could be possible to test like both of them separately and compare if they give you different signals, because well, with this DNA which is in solution, I think it should be a little bit easier to extract it, but with this on the minerals, you need to add some step to chemically release it from this minerals, and they may be maybe some difference in what we can see, and maybe like the resolution of signal may be different, because if it's in the solution, it can be much more easily mixed, and if it's adsorb on the minerals. So if there is no like huge bioturbation, minerals tend to stay more or less where they are. So this
Actually,
is just rough idea.
We did maybe a bit similar study. No, it's not exactly what you're saying, but we compared from modern sediments. That was surface sediments, so like upper 2 centimeters. So we can assume that here that most of the organisms are organisms like living organisms, and we analyzed the DNA from the bulk sediment. So we just took sediment sample as it is, easily the DNA and analyze it. And another part of the sample we sieved. We use 500 microns, 125, and 63 microns, and also we touched this fraction that is smaller than 63 microns, and analyzed these fractions independently to see how what would be the difference in the community composition and we analyzed forams. And there were differences in community composition, of course. The interesting thing was that these fractions of DNA, the DNA in this specific size fractions, more or less reflected the size of the forams. So we see like these big globothalamids in these bigger fractions, and a lot of these monothalamids, and these tiny ones in the smaller fraction. But the general trend was more or less the same. They were quite similar, surprisingly. But this is not exactly like what,
yeah. But like, it is also very interesting idea.
Quite similar approach, yeah.
Okay, so if I don't see any other questions, the last chance, last question? No? Okay. So Joanna, thank you so much for being with us.